And I'll do this very unslick maneuver to share my screen now. Okay. Yeah, thanks for the invitation to give the talk. And here we go. Hopefully we're getting there. Can you see that?
Yes, that's fine, Tom. Great.
Perfect. Yeah. So I'm going to talk about nitrous oxide emissions coming from agricultural sources, and in particular how we estimate those within the UK National Greenhouse Gas Emission Inventory. And the emission inventory is for the agriculture sector is funded by DEFRA, and the 6 organisations along the bottom in the central bit at the bottom there are the team that put together the agricultural emissions inventory, both greenhouse gas and ammonia emissions. So just what I'm going to talk about. So nitrous oxide from agriculture, what the process is, and just give you an overview of the current estimates. Then dig into the different reporting categories in a bit more detail. So we've got manure management, we've got emissions from soils and direct emissions from soils, but also indirect emissions coming from soils and from manure management. Then talk about how we come up with our emission factors for the inventory, which are a combination of sort of the IPCC guidelines, emission factors from 2006 guidelines. We've also looked at the more recent 2019 refinement to the 2006 guidelines, and as far as possible based on our own UK measurements. And then finally, just to have a quick think about what improvements we might make to the inventory. So emission processes. Now, when the nitrogen cycle in soil seems to have got a lot more complicated since I was at university, which admittedly was a long time ago. But there's now, endless sort of arrows between different forms of nitrogen in the soil, lots and lots of different processes, bacterial, fungal, and so on. So this is just one example that I pinched off the internet. You can see the citation there. So our understanding and ability to model nitrogen cycling is still incomplete because these diagrams keep changing and bits more keep getting added. And in terms of nitrous oxide emissions, there's lots of different competing processes which will influence how much substrate there is, how much how much the process will go down those different pathways. So very complicated and very difficult to capture in that sense and to predict. But from an emission inventory perspective, we've kept it much more simple. And in fact, this is the figure that we've put in the National Inventory report. So there's just one line really coming to nitrous oxide from denitrification. And so for UK agricultural soils, our assumption is that bacterial denitrification is the major pathway, and the key drivers for how much N2O is coming are the substrate concentration, so the amount of nitrate in the soils, and soil moisture. Just to have a look at the current inventory report, so the amount of nitrous oxide coming from agriculture in 2019 in the UK was about 50.3 kilotons, and we can express that as CO2 equivalents. That's using the global warming potential of 297. I think we still use in the inventory at the moment. We're using the IPCC assessment report 4 values. And that accounts for about 67%, so 2/3 of the total nitrous oxide that's coming from all sources or all reported sources in the UK. We can see that's declined. The agricultural emissions have declined by about 15% since 1990, and that decline is pretty much entirely due to reductions in livestock numbers and reductions in nitrogen fertilizer use. And we can look at the different sources within agriculture where those emissions are coming from. And so we've got about 16% coming from manure management, that's livestock housing and manure storage. Then we've got about 2/3 of the total emission is coming from soils in one form or another, direct emissions from soils. So 24% as from nitrogen fertilizer applications to soils, 10% or 11%, sorry, from manure applications to soils, 5% from grazing livestock, so the dung and urine returns, 13% from crop residues, 13% from mineralization of organic matter within soils. That includes organic soils, peatlands or histosols as we call them. And then there are indirect emissions, so they're coming from nitrogen that's been deposited, volatilized and then redeposited, or nitrogen that has leached. And so about 18% coming from those indirect sources. Yeah, sorry. And every year that we produce the inventory, this is also mapped on a 1 kilometre grid basis, the emissions. So this is work up at CEH, so Uli Dragasits and her team produce these maps. So you can see the left-hand map there, we've got manure management sources, pretty much maps on where the livestock, where we might expect to find livestock, so lots of cattle in the west, southwest, western UK, and some pigs and poultry in East Anglia. Soils emissions, wherever there's agricultural soils, so we can see that's pretty well spread across the country. The darker red bits that you can see just around East Anglia and slightly north of that, they are I'm pretty sure they will be the peatland soils, so where we're getting quite high emissions from cropping on organic soils. And then we can put those two together to give total emissions from agriculture. So if we go through those different categories that we report on, starting with manure management, and this does give us a little bit of a headache in trying to stick with the IPCC methodology and the emission factors that they define. In our model, our combined ammonia and greenhouse gas emission inventory model, we have what we call a nitrogen flow approach. So for livestock, for example, we start with what the animals are eating and how much nitrogen they then excrete. And that nitrogen can be excreted in different places, so on a pasture for grazing animals, in the livestock house. And from each of these sources, we will have losses of ammonia, of nitrous oxide, of nitric oxide and dinitrogen through denitrification. We may also have leaching if we get to the storage and the soil components, then we will have some nitrogen that is leached as well. And the remaining nitrogen from one pool will move to the next. So what we then, the manure we take from the house and put into a manure store is in this pool, and we can lose emissions there. Then we apply that manure to the soil and we have more emissions, and so on. And what we would like to do is to apply a discrete emission factor at each of those stages. So essentially for manure management, we say it's housing and storage of manures. We would like to have a different emission factor for each of those stages. Now, we've only got very limited UK data on direct measurements. We've got more on storage than from housing. So we need to look back at the default emission factors that IPCC give us for a lot of our sources. And the problem with the default emission factors is that they are for a manure management category that is a combination of livestock, housing and storage. So it doesn't fit very well with our model, and it's something we do want to improve. But we're living with it at the moment. In terms of the different types of manure, we know that slurries, liquid manures, pretty much associated with zero emissions. So that's relatively straightforward. But here's our table of emission factors for all the different manure types and sources. So we got the emission factor as a percent of the total manure N that is within the manure management category. We like to define some uncertainty limits around that, and this is where we got that from. So you can see we're predominantly using IPCC 2006 data. So for slurries, 0, or for a slatted floor house, which some cattle housing is, we do have a small emission of nitrous oxide. For solid manure systems, farmyard manure systems, where there's a combination of aerobic and anaerobic processes going on, we can expect to get some of the ammonia that's in the system nitrified and then denitrified. So we will get nitrous oxide emissions from solid manure systems. And we've actually made measurements of that. So we use our own UK specific emission factor there. So you can see we have emissions for the different types of livestock, so pigs, cattle and poultry. And we try to cover the different, so we try to cover the housing and the storage, but quite often we're including the storage component in with the housing because we can't segregate out the two parts because we haven't made the measurements in that way. Another thing that we don't currently do is take any account of the storage duration or the conditions of the storage, so the temperature, for example. So it's just an annualized emission factor. So if we say we have a farmyard manure system, the manure is in the house for part of the time, then it's put into a heap on the edge of the field for part of the time. That's it. We take no account of whether it's in the heap for 6 weeks or 6 months. So that's a definite improvement that we could potentially make if we have the data to do so. So moving on to the biggest category really, and that's soils, direct emissions from soils, and particularly from nitrogen fertilizers applied to soils. And there's been a lot of measurements on this. There was the large DEFRA-funded greenhouse gas platform project, AC0116, which generated a lot of data. But projects before that and after that have also generated data. Predominantly, those measurements are made using the static chamber technique that you can see in the top left picture there. So a large number of chambers put across various plots and treatments. We know that there's high spatial and temporal variation in nitrous oxide emissions from soils. So our sampling design has to take account of that. So we really do have a lot of chambers in the field, either multiple chambers on individual plots and also multiple replicates for treatments. And then a lot of sampling across the period of 1 year. Effectively, the inventory guidelines recommendations are that you should measure for a full year to account for emissions from the nitrogen fertilizer going to the soil. In fact, and you can see from the graph at the bottom here, this is typical of what we would expect to find. The majority of the emissions associated with nitrogen applications do arise within the first 3 months or so of that application. So here we've got the nitrous oxide emissions, these different coloured lines, these black bars at the top of instance of rainfall. And the arrows show where fertilizer was applied. So there were lots of different treatments, either different types of fertilizer or different application rates. So they were applied, and we can see, there's normally a signal, a response sort of fairly soon after the fertilizer application. And then sometimes we will get a subsequent response after a rainfall event. But once we get 2 or 3 months beyond that application, even when there's significant rainfall, we're not getting much in the way of emissions. So that's something that within all these projects, we did make 12-month duration measurements, but in terms of best deployment of resources, it really is worth focusing around these first 3 months and trying to get even more temporally defined emissions in the early stage. So we've got a huge data set then. So looking specifically at the different fertilizer types, we have ammonium nitrate. We had over 600 observations there, and Kirsty Top, SRUC, took all the data and did a regression and a REMO analysis on this, trying to see what the emissions were sensitive to. And for ammonium nitrate, we found it was a function of the application rate and the cumulative annual rainfall for a given site. And so a logarithmic function was derived relating that cumulative nitrous oxide emission for the given site and the given treatment to the nitrogen application rate and the cumulative rainfall for that site. And so you can see some examples here. The 3 curves give you 3 different nitrogen application rates, yellow being 100 kilograms per hectare, going up to the blue up to 300. So you can see the emission factor, which is on the y-axis here, increases with the application rate. So the proportional loss increases with application rate. And we've seen that before, that's understood, but also increases with the rainfall. And so we can see emission factors that we might expect across the UK going from, for ammonium nitrate, from 0.25 and a tenfold difference up to 2.5% of the applied air. If we look at urea fertilizer, which was the other fertilizer type for which we had a lot of observations, but not as many as for ammonium nitrate, this was found just to be a function of the nitrogen application rate. So we had a much flatter curve, in fact, in terms of the relationship with application rate. But we can see here the emission factor varying between just over 0.35 going up to 0.45 across different application rates. Soil texture, which we expected to be important as well, didn't come out quite as significant in the analysis. So using all that information and combining it with information on fertilizer practice, so we know the nitrogen application rates that go to different crops, and we know where those crops are in the country, and based on the cumulative annual rainfall for different grid cells across the country, we can kind of plot what we see as our emission factors for a given fertilizer type. So we can see there, because of the relationship with rainfall for ammonium nitrate, the emission factors will be much higher in the higher rainfall areas. But if we combine that with where the crops are, we will get the actual weighted average emission factor for fertilizer use in the UK. And our overall weighted average emission factor was 0.72% of the nitrogen applied. That's different. We can break it down into grassland. It was higher on grassland. It was a bit lower on arable. And then we can look at the individual fertilizer type. So ammonium nitrate was higher than for urea. But the weighted average was certainly a bit lower than the 1% that we used to use under IPCC. And it's certainly lower than the IPCC 2019 refinement proposal of 1.6%, which is recommended for wet climates, and most of the UK would qualify as a wet climate. So moving on to manure applications to land. So essentially we did the same thing, lots of experiments with manures, but not as much data as we had for fertilizers. But we tried to cover the different sort of representative soil and rainfall climatic types across the UK, looking at different manure types, so pig and cattle slurries, farmyard manures, and poultry manures being the main categories there. We tended to measure ammonia emissions. So you can see this is a small wind tunnel here. We could get an estimate of the loss by ammonia, as well as the nitrous oxide emissions using the static chamber techniques that you saw before. So there wasn't enough data really to do a full regression analysis. So we just took the mean emission factor, assuming that we'd done experiments across representative sites. And what we found there was there was definitely differences between manure types. So for pig and cattle slurries, we had an emission factor of about 0.75%. For the solid manures, it was much lower, 0.33%. And for poultry manures, it was pretty much 1%, which again, 1% is the default value for the IPCC 2006 guidelines. But 2019 refinement to the guidelines has proposes to reduce that 1% down to 0.6%, which I guess if you took a weighted average across all our manure types, we might be around about the same as that value. Moving on to grazing returns. Again, we collected urine and dung, and we did small plot experiments at lots of different grassland sites across the UK. And again, we made measurements of nitrous oxide and ammonia emissions. And we came up, we only used cattle urine and cattle dung. So these are the actual measured values here. So UK specific emission factor of 0.63 for cattle urine, 0.19%. So 0.63% of the N that was deposited in the urine was emitted as nitrous oxide, or 0.19% of the cattle dung nitrogen that was deposited. So these values compare with the IPCC 2006 values, for which cattle was 2%. So you can see we're much lower than the 2% that was given there. but we're broadly in line with the values that are in the IPCC 2019 refinement, which I think is not entirely surprising, because I think our data was a lot of the data sets that went into making that refinement. In terms of sheep, we've taken the IPCC guidance in that the values for sheep are about 50% of the values for cattle. So we don't have many measurements for sheep. We just assumed a 50% reduction compared with the cattle values. And again, we're broadly in line with the proposed 2019 values. The really important thing in differentiating between dung and urine. So previously it was just excreta, so the amount of nitrogen going in excreta, and you would apply an emission factor of 2%. If we know how much is in dung and how much is in urine, a potential mitigation technique within agriculture is to feed the animals less crude protein, to try and target the amount of protein they intake to what they really require, and then you'll have less excretion coming out, as and particularly less coming out in the urine, because most of the surplus nitrogen that the animal doesn't require comes out as urine. And so you can see you will have a disproportionate impact by having different emission factors for urine and for dung. And so we would actually see a greater reduction in nitrous oxide emissions than we would if we were just using a single emission factor to cover all of N excretion. So they were really big improvements that we made to the inventory there. Then we have crop residues. So these being returned to the soil, annual crops particularly, but also grassland renewal, which may be every 3 years, 5 years, or every 20 years. So we try to make estimates of that as well. Crop residues is a bit complicated. It's difficult to estimate. There's a lot of different parameters involved, and we don't have a huge amount of UK specific data for some of those parameters. But we follow the IPCC guidelines. For that, you need information on the crop dry matter yield, and we have good information on that for most of our crops in the UK. Then you need a ratio of the full plant above ground dry matter to the harvested yield. We don't necessarily have that. And similarly for the below ground dry matter, so the roots that remain after the harvest. Then you need to know the nitrogen content of those above and below ground residues, and the fraction of the above ground residue that is removed. Straw, for example, a lot of straw is baled and used in livestock bedding or for other purposes. And then you apply the emission factor. So for the amount of crop residue N that is returned, how much of that will be emitted? And the default emission factor is 1%. So for the UK, as I say, we broadly follow that. We use UK specific harvest index values in terms of the harvested yield versus the total crop yield, above ground crop yield. And where we have them, particularly for the major cereal crops, we use the UK specific values for the nitrogen content of those crops. But otherwise, we're using IPCC guideline default values for a lot of the parameters in those calculations. 2 minutes, Tom. Okay. So crop residues, this is just to show a lot of straw actually used to get burnt on the fields now and then ploughed in, but a lot is now removed because burning was banned in 1993. But there's a big question mark around crop residues. Are crop residues all the same? Well, we know they're not all the same. And the Minnow project, which was a few years back now, but did some experiments on this and looked at incorporating straw from winter wheat, so nitrous oxide emissions, whether the straw was removed or incorporated, and found virtually no difference in N2O emissions there. But with a green leafy residue, such as sugar beet leaves, they found a big difference in the nitrous oxide emissions if the leaves were incorporated, the residues were incorporated. And for crop residues, they sit on this line, if you like, of the amount of nitrogen and the carbon to nitrogen ratio. And the conclusions from that project really were that from things like straw, there's really no significant N2O emissions from crop residues, whereas from these high nitrogen, and they put a threshold of greater than 2%, there's definitely nitrous oxide emissions coming from those with an emission factor of about 1%. So should we look at this again and change our methods and how much we're estimating is coming from crop residues? Okay, so we're getting through these categories. Soils, mineralisation of organic matter in soils. Now, organic soils, if they're managed for agriculture, there's a lot of mineralisation. We know that. In terms of estimating the area of those organic soils and the emission factors associated with them, we have this really good report that Chris Evans led on up at CEH. So the maps, the peatland areas there, we need to then overlay that with the land use map to decide which of those are agricultural land uses, particularly cropland and intensive grassland. And in terms of kilograms per hectare, due to that land use of nitrous oxide coming from those areas, we've got emission factors 13 and 5.7 from those, compared with the default emission factor of 8. So in fact, again, the weighted average is probably not far off that, but at least we can differentiate now between different land uses. Mineral soils will also be subject to some mineralization if there's carbon stock changes in mineral soils, and this is estimated as part of the land use change forestry inventory. And if there is nitrogen being mineralized, then 1% of that is assumed to be emitted as nitrous oxide. And then remember, the last bit on that pie was the indirect emissions. So we have indirect emissions coming from N that is volatilized as ammonia and nitric oxide and then being redeposited. And we have our own UK specific estimates of that. We have very good estimates for ammonia emissions. We have less good estimates for nitrous oxide. We assume a ratio of NO to N2O, and Uta Skiva's done a lot of work on this, and you know, if we can get some more data, we can definitely improve there. But most of the N that's volatilized and being redeposited is actually ammonia. And then we've adopted the IPCC 2019 value, wet climate value, of 1.4% of deposited N will be re-emitted as nitrous oxide. Now that's bigger than the previous 1% under 2006, but we think there's the UK data that is available would justify using that higher value. We know that emissions are definitely higher than 1% on depositions to land that is non-agricultural, particularly woodlands, for example. So that's the value that we're proposed to use, in fact, in the coming inventory. And then finally, leached N. So agricultural N that gets into groundwater, into surface drains, rivers and estuaries. The emission factor we use for that, we do use the default IPCC 2006 value of 0.75. We've got our own estimates of how much is leached, and about 10% of N going to grassland is leached, 30% from tillage. And we reviewed what IPCC 2019 was proposing, and that was to increase that emission factor to 1.1%, but we didn't adopt that because when we pulled the data, well, I say we, this was mostly Stephen Anstey from ADAS, pulled the data set apart that underlies that value. The values for temperate Europe all seem to be very much higher than our own UK specific values. So we didn't feel we had quite enough values to really go ahead and have our own emission factor, which would have been quite low there. So we just opted to remain with the 2006 guidelines value. There was also some questions about how some of these values were put together, and digging into that Tiana et al. Paper. So I think there's a bit more work to do there yet as well. So that's a quick run through of all our sources, the emission factors we're using, the measurements that we've made. Improvements, yeah, there's definitely lots of improvements we can make. I mean, the inventory estimates these all as discrete categories, but obviously there's interactions between all of those nitrogen sources, and whether they're, you know, whether that has an impact on the emissions under different circumstances and conditions, you know, it's difficult to say. Improving our temporal resolution rather than just having an annualized emission factor might be useful. Using more process-based emission factors. We looked at using very good process-based models such as DNDC or DayCent, but I don't think they're not really fit for purpose for inventory use yet. That's for sure. better quantification of denitrification losses other than just N2O. Inclusion of mitigation techniques and nitrification inhibitors definitely come in there. Not used very much yet, but potentially will be. And then one for Alistair, understanding this gap between top-down measurements using tool towers, atmospheric dispersion modelling, and so on, versus this bottom-up approach that we use in the inventory. And there is a gap, and it would be good to know, are we missing something there, or are our emission factors not quite right? And I will stop there. Thank you, Alistair.
Thank you very much, Tom.
Try to find out how to stop sharing my screen.
Very comprehensive. Thank you very much. Time for one quick question. If anyone wants, before we move on to Yuri. You can always come back at the end, depending on time. Anyone got any burning questions? Oh, there's one. Matt.
Yeah, hi Tom, thanks. Sorry if I missed it during the talk. I've had kids here, but when you said at the end there that the process models are not quite ready for inventory work, can you just elaborate on that a little bit? What would be required to get them ready?
Yeah, so as I say, we looked at DACEN and UK DNDC or DNDC within the DEFRA-funded GHG platform projects. I think the problem was that you often need to tune them. If you look at a specific site results, you need to tune the parameters and play around with them a bit to get a good good match to what you're seeing on that site. And so that's one thing. And then you're extrapolating from that and how much tuning have you got to do across the UK. The second thing then was just the number of parameters that those models have, and that they're feeding the models. So the data sets you'd have to come up with. I mean, obviously we did, and then you're making lots of assumptions and generalizations probably in your data sets that you're feeding into the models. So at that time we felt that the semi-empirical approach, was more fit for purpose. But I still think that we can go further with the semi-empirical or the semi-process based approach without necessarily having going all the way to the UK, to sort of DNDC. Pete Levy is obviously making made a model, that's somewhere between the two, I guess, that it will be interesting to see to what extent we might be able to include that.
Great, thanks.
Okay, thank you very much. Okay, let's move on in the interest of time. Thank you very much, Tom. Much appreciated. So now we come to Yuri at PML, talking about marine emissions of nitrous oxide. Yep, we can see that.
Unmute myself. Yeah. Yep. Can you hear and see good?
Yeah, go for it.
Okay. Hi, everybody. So that's work that me and Gennady have been doing in the UK project, and as well as the building on work that we have done during the subsea biogeochemistry program. So I'm going to do a brief introduction about the marine side of the nitrogen cycle, and then show what are the driver of marine N2O production, as well as the air-sea fluxes. Show the uncertainty or some of the uncertainty that we have studied on the air-sea fluxes, and then try also to attribute which component of these air-sea fluxes are due to anthropogenic activity and which one are due to natural processes. So this is a simplified scheme of the nitrogen cycle. I totally agree and appreciate that Tom was already showing that how the nitrogen cycle in soil is very much a complex cycle, and is very much complex also in the marine environment. This is more a modeling point of view of the nitrogen cycle in marine water. And then in the biogeochemical model that is depicted here on the right, ERSEM, we've only actually also included only a small component, and this is the red part of the cycle. And so we only included in the water column, so in the pelagic environment, we included the production and then emission of N2O during the nitrification process. And for the benthic component is only the one during the denitrification process. This choice was also made because these were deemed the more relevant for the domain that we have studied. That is the Northwestern European Shelf. That's what we call the Atlantic margin model domain that has a horizontal resolution of about 7 kilometers, hence the name AMM7. And also, we particularly, I will focus in this presentation only on the emission and production on the shelf. That is the area contained by this black line, and this is roughly the area that is shallower than 200 meters. We run this the biogeochemical model ERSEM coupled to a physics model that is called NEMO, and that simulates the circulation and the temperature and the salinity of the marine environment. And to drive this model, we use atmospheric forcing, and in this case, we use the product from Urea Interim. And from now on, I will refer to this simulation with the name of Urea Interim because that's the main difference from the following one that I'll talk about later. And then oceanic boundary condition from global reanalysis. In particular, the river discharge and the nutrient load from the river was from a collected data set that was collected during the Shelf-Sea Biogeochemistry program, or SSB. And in this case, we simulated the 10-year period from 2005 to 2015. So this is the result of the simulation about the N2O production. So how much N2O is produced in the water column, and you can see that the production for the total shelf is about 50 kiloton per year, and for the UK shelf, that is the area highlighted here in blue. So the part of the shelf that is in UK territory, exclusive economic zone, is roughly a bit lower than half of it. But if we see in this 4 locations that we have highlighted here, we see how that there is obviously a temporal and also a spatial in the vertical dimension. And for those of you who have a marine background, can easily associate this to the dynamic of primary production. So basically, the high productivity areas, for instance, this one, but also this one down here in the middle of the North Sea, are associated to the decomposition of organic matter. So they follow the bloom when the phytoplankton is growing due to light, the warm water, and nutrient availability. And then when the organic matter that is also produced decay and then is mineralized, this frees a lot of ammonia that then is nitrified, and part of this nitrification process then is when N2O production occurs. So from both the map and this plot here, you can see how the more productive area are the region around the coast usually. But then if you look at how much the water can be saturated with N2O, we see a bit of the opposite trend, and how the Station B, that was, for instance, the one in the middle of the North Sea, reached very high saturation during autumn, and that's because when the water is sufficiently deep, like in this case, that goes down to more than 60 meters, then over summer the water stratified. There are lighter layer here on top. This line are the isodensity line. So this is a lighter layer of water that stays on top, and the bottom layer is not able, because of the difference in density, to basically exchange this N2O that is trapped throughout the year. And then when this stratification breaks up, then it's mixed over the entire water column, and only at this stage can actually be exchanged with the atmosphere. And this is indeed very evident if you look at the sea-to-air flux depicted here on the right. And we see that station A and B, that are the two deepest stations where the water is stratified. The largest part of the deflux happen toward the end of autumn, when the stratification breaks up, and all the N2O that has been accumulated in the deeper water can actually be exchanged with the atmosphere. While in the more fully mixed area, like the coastal area, station D, or in the one that are only weakly stratified, like station C, then the air sea flux is higher, basically at the same time as the production. The N2O, as soon as it is produced in the water column, that can be immediately exchanged with the atmosphere. And this is then the seasonal cycle of the sea-to-air flux, and it's basically summarizing what I've already said. And in particular, it is important to see how, especially in winter, when the water are cooler, then the sea can actually act as a sink of N2O, so it has a negative sea-to-air flux. And that's because in cooler water, the saturation increases, and so more N2O can actually stay in the water than the one is there, and so the flux goes from the atmosphere to the marine environment. And then if we want to compare these air sea fluxes of N2O with the emission factor, with the emission fluxes, sorry, that Tom was referring earlier on, if you look at the total shelf, the blue line, that is about 35 kiloton per year. And if you look at the UK only shelf, that's about 14, 15 kilotons per year, and are in, say, close to the emission from agricultural soil in case of the total shelf, or for fuel combustion for the UK only shelf. But how certain we are about these fluxes that we can estimate in order to understand? Oh yes, sorry. And the other thing is that the total production is about 52 kilotons per year, and only 35 kilotons per year are actually emitted to the atmosphere. This means that there are about 17 kilotons per year that are either accumulated on the shelf, or more likely are exported to the open ocean, where then this can eventually be exchanged with the atmosphere at a later stage. I was saying how certain we are about this estimate. So in order to understand this, we have done a different simulation. So the model configuration in terms of the two models, both NEMO and ERSEM, are exactly the same. But the main difference in this case is that we moved from the ERA interim atmospheric force into the ERA 5, that has different both spatial and temporal resolution, and also is an updated atmospheric reanalysis product. We changed the river reinforcing. Instead of using the SSP dataset everywhere, we used the long-term, large-scale model. That's a CH model from Vicky Beretol. You see the reference in a later slide. So we use that has a much better resolution of the freshwater discharge, as well as the nutrient load for the UK. And then we kept using the subsea biogeochemistry dataset for continental Europe. And then we extended the simulation period from 1990 to 2015. So here I'm comparing on the left the previous sea-to-air fluxes. In the middle is the new simulation, and on the right is the difference. And you can clearly see how the new simulation estimated much lower C2L fluxes almost everywhere, with only in some region the new simulation simulating actually a higher fluxes to the atmosphere. And if we want to summarize this difference in the big numbers, similar to the one we've seen before, this is the difference, and that's about a 35% decrease globally on both in the shelf and in the UK part of the shelf. So that's quite a big difference. And what is driving this difference? Why we are seeing this? So the first option is clearly because we're using a different atmospheric forcing. It could be that this new product ERA 5 has, well, it definitely has a different wind description. And so the ERA interim has a 3-hourly input and has about 0.70 degree resolution, while ERA 5 is hourly at the 1/4 of a degree resolution. And the plot in the map in the center show the average difference between the ERA 5 and ERA interim wind. And as you can see, there are some areas that show similarity with the difference in flux that you see in the small map on the top right. But in total, we can't really say that there is no real correspondence between the two maps in general. So the wind might be a component, but it's definitely not the major one. And so the new simulation that is forced with the ERA 5 has definitely simulated a cooler water column. This is the figure on the top left is the difference in the average difference in temperature, the surface temperature, and you can see that is about 0.2, 0.4 degree or even cooler in some area, and is also fresher. And part of this freshness is also due to the fact that we're using a different river forcing, so there is more freshwater discharge. And this, the combined effect of the temperature and the salinity gives that the concentration of saturation in the water, so how much N2O can actually stay in the water without outgassing into the atmosphere, is the saturation projected in the ERA 5 simulation is smaller than the one in the ERA interim, and we start seeing basically the same feature that we observe in the difference in fluxes, suggesting that this difference in saturation projected by the new model could actually drive the difference in fluxes. And so to understand that, we know that the flux, this is basically the equation of the fluxes, so that there is a factor that is driven by wind, that's the gas transfer coefficient. And then there is the saturation and the actual N2O that is projected by the model. And both for the wind and the saturation, we have the one that are projected by the ERA interim simulation and the ERA 5. So this is the first simulation that I've shown, and this is the second simulation that I've shown. But because we have all these different fields separately, we can also try to combine. So what would be the flux of N2O if we use the wind from ERA interim, for instance, but the saturation state or the concentration at saturation from ERA 5, and that would be the green bar that you will see in a second, or vice versa, the wind from ERA 5 and the saturation from ERA interim, that will be the orange bar. And this is the total flux on the shelf. The blue bar is the original flux in the ERA interim simulation. If we swap the wind with, so we use the wind from ERA 5 and everything else from ERA interim, we see that we only have a decrease of 2%. That is a very small decrease compared to the 35% that we have when we use ERA 5 both for wind and saturation. But when we swap, when we use the ERA 5 saturation and the ERA interim wind, then we say that there is the biggest drop, and it goes to a drop of 27%. And this is again another proof that is the saturation the difference in saturation projected by the two models that is driving the difference between the two estimates. Obviously, the two numbers are not yet exactly the same, and even if we factor in the wind component here, that is at least a 8% that is not justified by these two. And this can actually be explained by looking at the difference in the actual production of N2O in the water column. Again, on the left is the old simulation, the ERA interim driven one, and the middle the ERA 5, and on the right is the difference, and on the top here is the difference in the fluxes. And again, you can see similar feature, and here also the total difference that is about 11%. And again, this is so this difference is related to the changes in the net primary productivity. So the how much phytoplankton is produced in the water column in the simulation, because as I said at the beginning, the ammonia that is the main source basically of N2O is mostly produced as when the organic matter is decomposed in the water column. So which one of these two models can actually be considered more close to reality? Unfortunately, we don't have a lot of data of N2O in the marine environment. Here are the comparison. On the left are during the SSB program. There are different stations in the Celtic Sea during winter and spring and summer 2014-2015, and throughout the water column. The red simulation is the ERA 5, the blue is the ERA interim. As you can see, they both replicate quite good quite well the data, and there are not very much difference in this location. If you look at the spatial distribution of surface data, this is from the data here comes from the UKOA, so the UK Ocean Acidification Program in 2011. And again, it seems very similar pattern. If you look closer, there are some small differences, but they look very much similar, and similar for the saturation of N2O. If we expand and go beyond the simple nitrous oxide, and for instance, we look at surface temperature and surface salinity, you can see that, for instance, for surface temperature, the ERA 5 driven simulation is slightly better. If you look at salinity, it's probably the other way around. So all in all, there is no clear winner in a way, and so it is hard to pick one in terms of which simulation represents better the state of the marine environment. But how much of this flux is actually anthropogenic or linked to anthropogenic activity, and how much is simply due to the natural process that occurs in the marine environment? To understand this, we repeated the ERA 5 simulation, but we changed the nutrient load from the river, as well as the nitrogen deposition from the atmosphere into the marine environment. In order to decide how much we should change the load of nitrogen and phosphorus, we used this Bell et al. Paper, that is the same paper that presented the LTLS model output for the UK. For the continental Europe, we used this other paper that has done a similar study to the one that we are doing here, and this is the source for the nitrogen deposition. That in any case, this is a very small source of nitrogen for the marine environment globally in this area. So if you look at the difference, this time we're only comparing the ERA 5 with the so-called pristine scenarios in here and difference. In this case here, we only see a reduction of N2O emission to the atmosphere. And it's again mostly on the coastal area here, but also quite in a wide part of the southeastern part of the North Sea and in the Norwegian Trench. And then if we want to quantify, this is about 10 to 13% reduction, and in total number is about 1 kiloton per year is so the anthropogenic emission, let's say, from the marine environment, from the marine environment, or in the northwestern European shelf is about 3 kiloton per year, and if we focus on the UK shelf, is about 1 kiloton per year. And we can see how this that does not occur homogeneously throughout the year, but has clear seasonal pattern. And this is particularly evident in the more productive region, productive for N2O, so for instance in the southern North Sea or in the Norwegian Trench, where when N2O is produced, so generally is in the warmer period, late spring to summer, then you mostly see this difference. And then so it is actually, so this suggests that the reduced air-sea fluxes is basically due to a reduced production of N2O, and this is what this picture actually showed, that in the ERA 5 simulation, the total production in the shelf was about around 38 kiloton per year. If we remove the anthropogenic signal, that is about 33, and that's a reduction of about 4 kiloton per year that is produced in the marine environment. It's probably worth mentioning that when I say anthropogenic, it includes everything. It's not just the agricultural component that Tom was showing earlier, but it includes everything. Also the wastewater treatment plant, for instance, or industry, or all the other anthropogenic-driven nutrients that are leached into the rivers and then discharged into the marine environment. That's a lot, perfect timing for once. To conclude, the recycling of organic matter in the marine environment seems to fuel the N2O production on the shelf. The total emission from the UK shelf is about 10 to 14 kilotons per year, depending on which model we're looking at, and are comparable to the emission from fossil fuel combustion in the UK. As I clearly shown, these estimates are affected by a significant uncertainty, about 30%, and both the UK shelf as well as the global northwestern European shelf export about 33% of the N2O that is produced within the shelf to the Northeast Atlantic, and finally about 10% of the N2O emission and production from the shelf can be linked to anthropogenic source. And with this, I'm happy to take any questions. Thanks.
That's great. Thank you very much, Yuri. Excellent to talk about the whole marine environment around the UK, as the applause there is saying. So, anybody got any questions for Yuri? And you could start turning on your camera so we can see who you all are. That'd be lovely. Matt's got a question, but I'll just see if anyone else can come in before Matt. Has he got the other question as well? Go for it, Matt.
Yeah, thanks a lot, Yuri. It's fascinating stuff. So this, do we have any sense, this 10% drop that you saw in the total flux, was it flux or production? I can't remember. But anyway, that 10% drop, that's assuming a drop in anthropogenic inputs from the whole, from all the countries around the area, right? Do we have a sense for whether that's, so are we expecting a 10% drop for the UK? Could it be bigger as just the UK contribution? Could it be smaller? Do you know?
So I think that so the 10% is roughly for both production and emission, and I think that if you look at the total shelf, so at the changes in the total shelf, and then but only reducing the nutrient load from UK, then obviously this will be much less than 10%. However, we cannot think that a reduction in the nutrient load, the discharges from by the river from the UK, will only affect the N2O emission in the UK EEZ, because the circulation of the marine environment, a lot of basically all the riverine load that are discharged on the east coast of the UK, then are transported also outside of the UK EEZ. We can trace that with the models. We haven't done it yet, so that's something that definitely can be done. Another thing is that this scenario that we have run is not like a future projection in terms of nutrient management scenario. This is what if we were basically what if we remove everything that the human is putting into the marine environment? So for instance, from the estimate of reduction of nutrient that we took from the Bell et al. Paper was referring to 1800 type of emission from the river. So it's really what the aim is to quantify what is the anthropogenic fingerprint on that emission, rather than saying what is achievable by doing some nutrient management.
Thank you. Uta?
Can you hear me?
Yeah.
Good. I really enjoyed that talk. I didn't really know much about sort of coastal N2O emissions. So how do you measure these? I mean, do you just sort of look at the nitrogen contents and calculate the emissions from there, or do you do some actual measurements from the boats? Or
so 95% of what I've shown was model output.
Yeah.
So in terms of the measurement, I'll start something, but then Andy, who's in the call, can answer much better than me. But basically, so the N2O concentration are measured taking sample of water and then brought in the lab, and then Andy will fill out the measure. I haven't used any direct measurement of air sea fluxes here, but people in our in PML can also do direct or they measure the air sea fluxes using the covariances method.
Okay. Mm-hmm.
But I haven't used those in here to ground truth the air sea fluxes.
But from what you said, it sounds like you need many more data of N2O emissions, proper N2O emissions using eddy covariance. Probably.
But both from the eddy covariance, but also from the N2O concentration in the marine environment, because then because if we only look at the observation of the air sea fluxes, then we can constrain that process, but we're not 100% sure that the production process is well constrained. So if we can have both, as always, more data, the better it is.
Yeah. Thank you very much.
Shall I just add a little bit to that? And it's not a lot more, but essentially it seems like myself and my colleagues have been hiding data from Yuri, so we've got I think a lot more measurements than he seems to think we have. We often go to sea and measure them on a ship. All around the UK and throughout most of the world, we've made measurements. We don't currently have the capability of doing direct flux measurements by eddy covariance for nitrous oxide. We do for CO2 and methane. N2O is something that we're working on for the near future. And we've got lots of coastal measurements as well. So we've got a local time series station where we've got about 15 years worth of weekly, or there's a few gaps, but and quite a lot in our local estuaries as well.
Yeah, just one thing indeed that Andy picked up is that everything that I've shown here is because the resolution of the model is about 7 kilometers. That's really outside of estuary and also not really coastal, because one box is basically a homogeneous box of 50 square kilometers, so it's very large. And from the work that Andy has done also within the UK, a lot of emission also happen in the estuary, where there are high fluxes, but because then the surface of this estuary tend to be small, then at the global level or at total UK level, that adapt to a less significant flux compared to the one from the entire shelf, where the surface is much larger.
Great, thanks. Matt, you have another question?
Yeah, I had a question for Tom, really. I don't know if it's possible to answer it, but I'm thinking about the difference between the top-down estimates that Alistair comes up with for the UK and the inventory. And it seems like Alistair's estimates are always slightly higher than the inventory. Is that right, Alistair? That's the right way around, right? Yeah. I'm wondering if you have a sense for, like, what would be your main guess, your best guess as to why that might be?
Yeah, I've always said in the past, my feeling is something to do with wetlands or estuaries, or, you know, I don't know what the estimates from those areas are, but there's quite a lot of N going out of the agricultural system, you know, in water, and it is a very uncertain one. If you go back to the 1996 IPCC guidelines, that emission factor 5 for leached N was 2.5%. Then that came down hugely, down to 0.75%. Now it's proposed to go back up to 1.1%, I think it is. You know, so that just shows how uncertain that number is. We've got our own UK, so I've forgotten his name now, University of East Anglia, who's done some measurements, you know, fluxes from water in drains and rivers and so on. So we've got some UK measurements which suggest it's low, but that's kind of in the water course as it's going. But I just think there's a lot of N probably deposited then into wet areas that you know could potentially account for this. But I don't know. You know, I think that the thing is, as I said before, the way we do this as well is sort of discrete boxes that we're estimating from. You know, there's uncertainties in all of those, and uncertainties in the sort of synergies, and that's where it's really interesting to start putting eddy covariance measurements alongside bottom-up type measurements for discrete sites, and see if we can get a bit more understanding around that. But I was going to ask Alistair the same question, really. So, to what extent are these ocean fluxes included in your names model? Because the UK estimate was about 10 kilotons or something, was it 10 or 12? You know, that's quite a big chunk. But you know, I don't know how much of that is accounted for in what you're measuring from the tall towers.
Yeah, so in the tall towers, I would include them because I include the UK seas as well as UK land. And my question actually was back to you and Pete, really, was to say that how much of that actually is covered by the inventory? Because I know you get some, obviously, conversion rate, but what do you think? I mean, do you think that ultimately that's a low number if you compare it to Eura's 10?
Yeah.
Have a feel for whether it's 1 or 2 in the UK in the inventory, and possibly should be 10 when you've gone the whole length of the river out to sea. Yeah,
so the indirect from leaching was about, if you said it was 10%, so perhaps it's of the order of 5 kilotons. I think I'd have to double check and look at that again, but yeah. so it may be an underestimate. Yeah, I don't know to what extent we factor in what's coming out in the oceans as well.
But can I just a point of clarification there? I think Yuri, that 10, that's mostly natural, isn't it? It's actually a much smaller
Yes.
number if you compare Yuri's pristine and.
That's true. Yeah.
Yeah,
It's
so
10%
the anthropogenic
of that
component that is emitted in the UK EEZ is about 1.
So actually, so Alistair, you would be getting in your inversions, you are getting that big natural component then, essentially, right?
Yeah. So I mean, I was quite surprised that the reduction was only 10% when you remove all the human input of N on the land. You only get a 10% drop in emissions.
Because a lot of the nutrients in the shelf come from the ocean, from outside, and not from land.
Not from okay. Well, okay, so it's not. Yeah.
And also the model is not simulating anything that happens in the estuary, where the anthropogenic signal will be much higher than in the wider shelf, where there is a lot of the influence of the open ocean.
So is there any way for me to remove the natural component and just retain the anthropogenic component then? I mean, do I go too far out by using the whole of the UK sea area?
Well, could you just subtract Yuri's pristine fluxes from your total?
Could do. Yeah, that would be easy enough because we can grid it. Presumably, Yuri, do you provide both those numbers to Pete? You haven't yet, have you?
I have only given the one with everything, but I mean I can easily do exactly the same thing, and so then you can subtract one from the other to have only the anthropogenic signal.
An interesting starting point, wouldn't it? Yeah.
Yes, because that might knock. I mean, I think we'd still end up that I'm estimating higher than you, Tom, and whether it's my model or emission factors it's definitely an open question. But yeah, that would be definitely a good start.
I think one problem with the
Appreciate
comparison
how
is,
big the natural component was.
one problem with that comparison is that the ocean term is quite a big number, but it's very diffuse. It's spread over a very large area. So compared with the land fluxes, they're quite small in the instantaneous terms.
In what you're
Quite a hard
measuring
signal to pick up, I suspect.
Yeah.
I think it's I think it's this estuary component which is probably missing from the consideration. So that the amount of N2O that hits the ocean from the estuaries is quite small. It seems to be lost to the atmosphere, or it's denitrified within these estuarine systems. So there's a big consideration to take on here, I think.
So do your measurements support that there is a potentially like nationally significant source from the estuaries then?
Whether it's nationally significant, I suspect it probably is, but we haven't done the numbers on that. We're just working up some fluxes for. We've got our locate project running at the same time, which has been doing estuaries, and we've focused mainly on methane. But over the next few days, I'm going to come up with some nitrous oxide numbers for about 7 or 8 of those estuaries. So a couple of days' time, I might be better informed to tell you what they look like. But yeah, it's certainly we get a big peak of N2O for all of those estuaries in a kind of mid-estuarine environment. So it seems to come in, and the processing in the estuary allows more of that N2O to be produced and released, and by the time it gets to the ocean, it's come down. So we don't see a big export from most of those estuaries to the ocean.
Interesting.
Very interesting to